Hello, everyone, and welcome to the second in a regular series of seminars on sea turtle medicine hosted by the Sea Turtle Rescue Alliance. I am Dr. Max Polyak. I am the Director of Rehabilitation and the attending veterinarian at Loggerhead Marine Life Center here in beautiful Juno Beach, where I'm joining you from today. We have a really terrific guest that we're all very excited to introduce to you and to hear her chat. But before we get to that, I just want to give you a little bit about the background of the Alliance and a little bio of our guest speaker today. So the Sea Turtle Rescue Alliance is a fairly new initiative that really has as its foundation increasing the access to specialized sea turtle medical care around the world to places that don't normally have access to that. That's a critical part of our mission, which is directed primarily in the conservation of these wonderful species that we all love and we treat and we study. A second part of our mission is to foster a collaborative environment around the world among medical professionals, biologists, government officials, and citizen scientists in improving the overall health and well-being of sea turtles worldwide. So with that said, we are really happy to introduce to you our guest speaker today. She is Dr. Mary Escobedo Mondragon from Mexico. She is a wildlife veterinarian who graduated from the Autonomous Metropolitan University. She has a graduate degree in wildlife medicine and management from National Autonomous University in Mexico. And she is furthering her graduate studies there to the PhD level. She has over 7 years of experience working and studying sea turtles in the field and in a medical environment in North America, in Europe, and now is an active member of a number of international organizations committed to improving the welfare of sea turtles. Among those are the Sea Turtle Rescue Alliance. She's also on the Sea Turtle Committee in the Quintana Roo state in Mexico. She's also an active member of the Mexican Association of Turtle Veterinarians. So today, we are delighted to hear her expertise in the field of sea turtle biomonitoring in the Quintana Roo state of Mexico. So everyone, please welcome Dr. Mary.
Thank you. Thank you for the invitation. I am very happy to be here with all of you and sharing a little bit of our work that we do in Quintana Roo state. So welcome everyone. Good morning, evening, afternoon, night probably. So thank you for coming with us to share all this information. I am very happy, and as you say at the beginning, Max, veterinarian, it's a very big field, and we are going to see just a layer of our work as wildlife veterinarians, because wildlife veterinary have a lot of layers, and we can work in a lot of fields inside wildlife veterinarians and of course sea turtles. So I'm going to share my screen. So is it running?
Yes.
Okay, thank you, Alex. So hello, everyone. I'm going to talk about this, that is Sea Turtle Biomonitoring in Quintana Roo State, Mexico. My name is Mary Escobedo Mondragon, and I am from Mexico. So biomonitoring is defined as the act of observing and assessing everything that is ongoing and happen in the environment. So before talking about it, I'm going to talk a little bit, how do I get into biomonitoring? So as Max said in my introduction, I am a veterinarian graduate from this university that in Spanish is Universidad Autónoma Metropolitana Unidad Xochimilco, or in English, Autonomous Metropolitan University Xochimilco Unit. And during my social service, that is like the last part you do while you are as veterinarian or in any profession, I start working with sea turtles in Oaxaca state. In there, I help with a lot of areas, even during the turtle camp and nesting season, but also I was start learning and doing research in biochemistry and hemathology in sea turtles in Olive Ridley, and also helping with disease in the rehab center of the area. After that, sorry. Oops, sorry. I'm going to go back. I think I can. Well, after that, I do my master degree in biomonitoring, and that's how I go in through this project. So what is biomonitoring? Biomonitoring is defined as the act of observing and assessing the state and ongoing changes in the ecosystem, components such as chemicals and their species. So the environmental monitoring has been considered a valuable tool for both ecological and human health surveillance. And it's very important to, in all the ecosystem and habitats, we do biomonitoring to track all the issues and probably concerns about contamination and pollution in the area. So the most important part of doing biomonitoring is to be very cautious in the interpretation of the data, and also it's one of the most sensitive issues in this type of analysis. So why we choose sea turtles for that? Well, sea turtles are incredible animals. They are a long-lived reptiles. They have a very important life history that teaches about two different habitats, that is the terrestrial and the aquatic habitat. Also, every species have a very particular diet, so we can see all these different things between species and how they move through their environment, through its diet. So we can study the bioconcentration of the chemicals, the bioaccumulation, and the biomagnification. So all of those components help us to understand the biomonitoring studies and use sea turtle as a bioindicator, also sentinels of the sea. So this is two charts. We took these charts from Bolton, and also it's in the book of Manier that was published, I think, in 2017. And in here we can see three different species, that is the green turtle, hawksbill turtle, and loggerhead turtle. So as we can see, they have different life history, but green turtle and hawksbill turtle share life history, but that is not totally true, because the big and huge difference that we are going to see in the life history of both these species are the feed zone and also the diet they have. So it's different, and that makes them unique, and that's the reason using different species for biomonitoring, it's very useful to understand what is happening in different areas, even in the sea. It's not the same studying green turtles that help us to visualize and analyze what's happening in the seagrass. And also it's very different with hawksbill that help us to understand what's happening with the coral reef. So what are we doing in our sea turtle biomonitoring project? What we do is all these topics, and we study the toxicology, specifically of inorganic pollutants. We also study the strandings, and we do forensic medicine. We focus in majority in dead turtles in strandings. We do pathology, parasitology, and molecular biology of infectious disease. And why we choose Quintana Roo? Well, we chose Quintana Roo because it's an area with a high biodiversity. As you can see, this is Quintana Roo, this is Mexico. Quintana Roo is part of Yucatan Peninsula, formed by 3 states, that it's Campeche, Yucatan, and then here we got Quintana Roo state. So this side is Quintana Roo, and all this area is highly biodiverse of marine wildlife. So in there we have all these species, that is green loggerhead, hawksbill, leatherback, and Camp Ridley, as a feeding area and also for nesting of major in these 3 species, and we sometimes have leatherback too. So in all this area we have nesting beaches for green loggerhead and hawksbill. So as I said, we mainly focus in stranding, dead turtles, and nesting. And for nesting, we are studying green turtles, loggerhead, and hawksbill for this study. So what we do in fieldwork, we're going to start with the part of the strandings. So we do sections or necropsies to understand what is happening with the population. So as you can see in these photos, we are doing different things and also in different areas, and depending on the place, that's the thing that we do is not the same. Found this type of turtle that it's a carcass and is like a fresh dead turtle, but also as you can see in here, we are doing a necropsy in field, and in here we have the possibility to have a veterinary area to work better in doing all these dissections and necropsies. But it's not always easy to find these locations when you work in wildlife. So we have to do also this kind of work, that is doing the necropsies at field. But also something very important is that Quintana Roo state, it's a large state. So it's impossible for me travel to all the state during all the years. So sometimes I'm not in the state, sometimes I am far away where the stranding is happening. So I have the help and assist of many people that is helping to work out this project. So one example is in here, that what happened, I was far away from the stranding. So the people of Isla Mujeres help us, and they buried the turtle, and at the next day with the help of Tortugranja help us to do the necropsy. So I travel, and at the next day we have the turtle to do the necropsy at field. And in here it's in the National Park of Puerto Morelos Reef, where the biologist Erika is very commitment, and she have full time dedicated of the project of sea turtles, and also in our project she's wonderful. She's been helping us a lot. So what she do is help us with the necropsies that happen at the national park where she works, and then I travel and we start to process part of the tissues that she took for sampling, and also we package everything as supposed to be preserved, because it's not the same conserve the samples for toxicology studies than for molecular or genetic studies. So we need to do it everything right so we can do all the studies properly. Also at the field work, when we're working with the nesting turtles, what we do is have a quickly physical exam to see that the turtles are healthy. We took some biomeasures to see how big they are, if they have something in particular, the tags they have. We record all the tags in the beach to know if they have been there before or not, and all the management of the turtle camp needs, we help them too with that part. But also in here, as we can see, we are taking the measure of a tumor. We didn't take a sample for this because it's not right now, it's not part of our project. Now we are working to add it to our project to analyze the possibility of finding herpes virus, but this is another story. So in here, we are watching how we are taking a sample of the scute of a nesting turtle. This is a green turtle, to later analyze it in our lab for toxicology. And in here, after all this work at night, at day, we put a laboratory at the turtle camp we're working and start analyzing the blood samples to do all our parameters and see that turtles are healthy. So in our laboratory work, we have to do a lot of things. Here it's just one of all the things we do for biomonitoring. This is the part of toxicology. And we have to clean all the tissues because we took also tissues for toxicology for the stranded turtle. And as you can see, one of those was a carcass. So we have to clean very well all the tissues that are exposed to the environment. So we clean it perfectly to only be measured what is inside the tissue and not in the environment. So in here is already the sample process for running through the ICP. That what this equipment do is read all the pollutants, inorganic pollutants that could be found in the tissue. And also we are support and we collaborate, and sometimes it's just a service. We took the samples over there into different institutions and also different laboratories. So we want to thank all of the laboratories that help us and support us to do this project. And so this project starts in 2017, but we start taking samples since 2018. So we've got 4 official years working in this project, and we have published, these are the last articles that we published. This is from this year. And we published from all the findings that we got in field for the stranding and for the nesting turtles. So we're going to talk a little bit about it so you can see what are our results and what are we finding in this biomonitoring program. So in this article, basically what we found that it's already have done in other species and also in some turtles, not maybe all these groups, not maybe all of these pollutants, maybe just 1 or 2 groups, but still it's the same conclusion that scute, it's a very good tissue to do biomonitoring because it allowed us to understand the bioaccumulation, the biomagnification, and also the biodilution of all these components that we are going to find in sea turtles. While blood, it's a very good tissue for bioconcentration. So it allowed us to see what is happening in the moment, while the scute helped us to understand what had happened before, like a couple of months or years before we encounter our sea turtles. What we are working now is in the comparison of the 3 species and how all the diet, the traffic change, and all these traffic levels influence into the different species of sea turtle in different areas. And right now we are starting to add in the south part of the state. So in all these 3 species, we are taking blood as well and scute in the nesting turtles, but also now we are starting to analyze and see the effect of the blood cells between those pollutants that we found and the health state of these sea turtles and the pollutants that we are finding, if they are correlated or not, and influence in between the health of sea turtles, in nesting turtles in the state. Also, we are working in the part of the stranding. This is something that we hope that it's being published at the end of this year or at the beginning of the next one. It's been a little bit challenging because of the pandemic. So we hope that this could be published soon. So we are analyzing all the data that we have of the strandings, the interesting findings of those necropsies, stomach content, the incidence and prevalence of some disease or the type of stranding species, season where we have a high tendency of having strandings and where we are having strandings. And also we are trying to do in a couple of months some modeling that help us to understand if environmental changes have a direct effect into those strandings. So we hope we can publish that too and find something interesting. This is also a little bit of what we have found in the strandings, and there are interesting cases like this of pathology studies that we have done and about phyloepomycosis in free-ranging hawksbill turtles. And this is very important because these findings help us in rehab center to understand how to treat and how to manage this type of disease. So even if we are not working in this project in a rehab center, we're helping to give more data to give the correct diagnosis for this kind of disease and also how to treat them and what kind of analysis use when we think that could be this kind of lung disease in infection diseases. We also have worked all this year in workshops. So we have workshops for many institutions. This is just a few, but we have done many in different institutions, even in our university for graduate students. So we help them and teach them how to do necropsies, how to do all these steps for following a correct necropsy, what we have to do in field, the findings that maybe we could see and help us to understand the macroscopic analysis during the necropsy, and if they are related or not with human impact. That is something very important to find while we are doing a necropsy, and it's something, a very high concern that it's immediately to attend in at least at our state and our country, because if we find a turtle that have died for a disease, it's not the same and it's not the same management that if we found a turtle that had been hunted or had been poached or had been an accident with a boat strike, because that requires an immediate action. We also have veterinary externships. This is some of them. We also receive biologists. So you are welcome to come and help us to the biomonitoring program. As you can see, I am very happy to say that most of our veterinarian externship are women. So I am happy to share this little bit of the work these girls have done. We also have, of course, men that come with us, but the majority are women. And also, again, I want to give an acknowledgement of all these institutions that help us, because this project, it's a project of all the state, because we require the help of all of those institutions that have some management and are a part of these protection and conservation programs of Quintana Roo state. Most of them, I think, yes, most of these institutions are part of this institution, that is the Sea Turtle Committee of Quintana Roo. So thank you very much also to the committee that it's been supporting for all this year the project. But we also have new ads, and that's the Mexican Association of Sea Turtle Veterinarians. Here you can go through the web of AMVT and find a little bit more about the project. I summarize as much as I can because we only have 30 minutes to explain a little bit and to share a little bit about this biomonitoring project. And this is association. It also have different projects, but something that is very important, and the reason this project now is also part of the Mexican Association of Turtle Veterinarians, is because it's found in the part and of research and scientific publishing. So if you are a veterinarian that is highly interested in doing research, this is a good opportunity to go through this association and also to share all the information you are producing. And as I told you, they have 6 ongoing projects. One of them is biomonitoring project. The current allies are these 6 ones, because these both are together. That is Menos Plástico, Fantástico and Mahahual and Sandy Turtle, Camp, they are together. So we have these 6 allies, and now we are happy to share with all of you that we are going to start a partnership with Sea Turtle Rescue Alliance. So we are very happy to be here with you sharing this project, but also sharing this partnership and helping with all this part of assessing and sharing information in Spanish for Latin America. So thank you for the invitation and for this partnership. So thank you very much. I give them in here our email contact if you want to find out more about the project or how to get involved with us in any other project. So again, thank you.
Thank you, Dr. Mary. That was terrific. A really fascinating talk about a very, very important part of the world related to these species. And we couldn't be more thankful for your insight and for sharing it with us. We do have a number of questions, and I hope you can stick around for a bit.
Yes.
Okay. The first one is, your externship sounds brilliant. Could you tell us a little more about this, please? How long do you normally have externs? And how can is it open to veterinary students and graduated veterinarians? So a couple of questions kind of rolled into one.
Yeah. Okay, so the first one, it will be, let me see if I can find it so I don't forget about the question. I don't think I can find it. So the first one was how to get involved, right? So as you can see in the last slide, we've got our contact, but also you can go through our webpage and ask for being in our program. You can also contact us through Facebook of AMVT and to ask for participate in our project. In biomonitoring project, what we are working is with different areas. You don't have to be veterinarian. You can also be a biologist for this project. So you can come and be with us, but depending on how prepared you are in your studies, it means if you are in the middle of veterinary career, we are going to probably ask you to wait a little bit more, but because as you saw, we required very trained people. So it's also very intensive work at field. We do field work sometimes just for 1 month. So in 1 month, we have to go out all night to have all our samples and also to analyze. So it's very intensive. So we prefer that the students that come with us are graduated students or at the last year of the career. So if you are a veterinarian, you must be in the last year of your career. And if you already graduate, you are more than welcome to come with us. And I think it was that, or there's another question inside that question.
That was a great answer and very informative. And it sounds like a really exciting opportunity for the students out there that want to work on your important projects.
Yes.
Our next question is, what is the most common disease that you see in your state?
Well, it's not actually infectious disease. What we have seen in the strandings is a lot of those strand turtles came because they are debilitated. So it talks about how the environment it is. Maybe they don't found food, and when they come to the Caribbean and finally find food, sometimes they just have a, I don't know how to say it properly, like a party of food, and then they don't find the correct food of their diet. So we got, for example, a case where a loggerhead was very emaciated, but the entire stomach and intestine were full of puffer fish. So all those puffer fish gave them a big damage of this turtle, and that turtle died because of that. So most of our stranded turtles, dead turtles, have issues with debilitated state. So they are emaciated. So that's a big concern. We're trying to figure it out, what's happening. It's very strange that we find infection disease, and the infection disease that we are finding are lung disease for infection, fungus and bacteria. And also we are finding a little bit of troubles with fibropapillomatosis, FP. So that's the reason this year we are happy to say that we are starting a project in collaboration, in partnership with another institution that is working with that. So we are going to start taking some samples to understand what's happening in the disease dynamic of this concern of fibropapillomatosis, FP.
Wonderful. That was actually a follow-up question of one of our viewers about PAPS, and it's exciting to hear that you're going to investigate that further. It's interesting, the chronic debilitation or the debilitated state that you're seeing there. We see that fairly regularly in our waters as well, and not the same acute presentation where the food items are causing potentially some of the presentation itself, but the chronicity of it is related to the food supply, at least on a certain level. So really interesting. Another question, what has been the discovery from your biomonitoring program that you found the most interesting over the course of your research so far?
Well, for me, it's finding electronic pollutants. So at the beginning right now, we are with the part of bioconcentration, biomagnification, biodilution, and bioaccumulation. It means in the traffic change, how those pollutants had been found in the different species related to its diet. So one of those pollutants are all these groups that are rare earth elements that most of the source are electronic bad management into the sea. So it's a very interesting area. It's a new, I don't know what's the correct word, like a new site inside biomonitoring studies, because all these chemical components are relatively new. So we do not understand how fast they travel into the traffic change. We are start studying the nocive effects of those pollutants, and also to start regulate how we dispose these pollutants. So it's very interesting knowing that right now there's a huge concern about those electronic pollutants and what we need to do a correct disposal and environmental management for that.
That's wonderful. Now, a kind of a different question that that people are beginning to review related to the pandemic. And a question we have is, did you see changes in frequency or type of strandings during pandemic compared to pre-pandemic and now sort of post-pandemic?
Yes, actually, that is going to be also in our publishing because it was very interesting. I don't know if we got more people observing those issues or it increased, but we have an increase of poaching. So there are not true strandings. We call them pseudo strandings because the turtle is just dead in the beach, not because she couldn't return to the water, to the ocean. It's because they get poached. So it's a huge concern that we are searching and researching to know if that was a specific event of the pandemic, or it's just we got more observers reporting that because we've got more time to do the monitoring area, and also in the south, because in the north we have an active monitoring, but in the south we start like 2 years ago. So also it's something very new. So it could not be related with the pandemic. So we are researching that.
Excellent. That's going to be fascinating information when you report it. The next question is plastic ingestion a common phenomena that you see on your postmortem exams?
It's been increasing. I actually, at the beginning of the research in 2017, we didn't found any plastic content in sea turtles, but in the last year, we start finding those plastic contents. So we start seeing that, but they are not a cause of death. We just found they have eat a little bit of plastic. So right now it's not a huge concern, but we are seeing that tendency of eating plastic and marine garbage is more common in find those in our turtles that strand in the area.
Okay, interesting. Now, we kind of talked a little bit about the most common disease that you see in your state. Linked to that question, what's the largest threat to sea turtles that you have found in your research to date?
I think it's the food source. I am very worried because it's not as a disease. It's just, it's not a high quality food in the area. And that also have an impact in other species. But because also what we do, not all the time, but we do some monitoring in the water, in the reef barrier. We got the second biggest reef barrier in the world, that is the Mesoamerican system. So if turtles are not finding high quality food in our feeding area, that it's the Caribbean Sea, then that's the reason they are also getting sick of maybe other disease that we are not seeing, because they suffer quickly the loss of food, the quality, and also that's the reason they are coming in debilitation state and also emaciated, because they are not finding food. And I think for me that's the huge concern of the area more than disease right now. And talking about disease is not for the stranded turtles or from the juveniles. The concern right now in juveniles are fever papillomatosis. We have seen, it's not my project, it's another project that is other people working on that in the region, that they have seen an increase of the presence of this disease in juvenile turtles in the region. So also in disease, that's a huge concern because it was not that high.
That's interesting about potentially disease in the food supply itself related to the quality and the scarcity of it. Kind of linked to that point, have you seen any influence from the recent oil spill in the Gulf and any effect on your toxicology that you are running on specimens that you have, or anything kind of related to that type of an environmental exposure to these species?
Well, right now, in this moment, we didn't see immediately effects of that. Probably in a couple of months, we're going to see it. Right now, that I know we don't have any report of that, but during our study in these years of biomonitoring, of course, we have seen some pollutants related with oil spills and also with pollutants of this discharge in the sea in the Gulf of Mexico, because Quintana Roo also share a little bit of Gulf of Mexico. At the north side, we've got this point that share waters with Caribbean Sea and Gulf of Mexico. So the current doesn't go down through the Gulf. What does the current of the Caribbean goes inside of the Gulf. So it's what we have seen, it's just a little bit of those pollutants of the Gulf. It's not too concerned about it, but we have seen it in the pollutants that we found, that we've got a little bit higher some components that are related with all those organic and inorganic pollutants of spills.
Interesting. Those are interesting details about the nuances of the currents there too. We have, I think here, one final question that is more related to the organization in Mexico. Can you tell us a little more about the Mexican Veterinary Association and the turtle component of that, and how people might be able to get involved more if they are in the area or if they're around the world and want to come and help?
Sure. So the Veterinarian Association was founded by wildlife veterinarians that work with sea turtles. So we are all around the country working with these 6 different projects. And what we are doing is working with rehab. We have some area that have the medical, the medical area to work with the rehab turtles and to treat them. And that center is in the Gulf of Mexico. We also have in the north side of Baja California another project that is a health assessment project. And over there, they are also looking for all these health and disease problems. They are starting to focus also in fever papillomatosis. And that's one of the projects that we are now start sharing information and also doing partnership and collaborative things to find out what is happening in the both sides of the country, in the north and in the south. And also we have a partnership at the south side of the Pacific Ocean of Mexico. And in this part, they also work in wildlife, and they are starting doing his health assessment project. So the component of veterinarians is that the ones we are working at field, we are making a baseline for diagnose disease and also to treat them. And in the ones that are working in the rehab center, they are start doing and recording all the proper techniques and management for different situations that we are seeing. In this case of the Gulf of Mexico, in the Gulf of Mexico, we have a high impact of human impact. So we are start working on that. And you can find more information of how to get involved in our webpage, that is amvt.odoo.com, or also in our Facebook page, that is AMVT.
Terrific. Thank you so much. We are just about out of time here, and I think one of my colleagues is going to join us just for a signing off, but I would like to be the first to thank you again. And it was a fascinating talk and really amazing work you're doing in that part of the world. And kudos for the initiative in Mexico and what you've been able to accomplish there. It's really, really exciting. So we look forward to having you as part of the Alliance, as an active member, and as part of the regional structure of the Alliance in that part of the world. Really, really happy to have you aboard. But we are going to also have,
Thank you.
You're welcome. As I mentioned earlier, that this is a regular speaking feature, and we want to do this, and we're planning to, on a monthly basis. So we're excited about that, and excited about sharing the work that colleagues are doing around the world in sea turtle medicine, biology, and conservation. So next month, it's scheduled for Monday, August 16th at 8 p.m., which is 2 p.m. UK time, 8 p.m. in Thailand, is going to be our next speaker, who is Dr. Wachara, who is going to be speaking on sea turtle rescue in Thailand, the status, case studies, and key players. So we're really, really happy about our next talk, and we hope everyone will join us. I think I'm going to be signing off, and another Alliance steering group member is going to chime on just to wrap things up and provide any more details about this talk and the next talk. Thanks, Mary. Really, really great seeing you.
Okay, thanks. Thanks to you too, Max.
Okay, bye-bye.
Bye-bye.
Thanks, Max, so much for hosting, and thank you again, Mary. That was an incredible talk. It's just so wonderful to see your work, how much you do out there in Mexico. You're a one-woman band and women's science. I'm really proud of you and your work. So grateful to have you as our second speaker. And as Max said, we are obviously going to move the medical series forward next month with Dr. Wachara from Thailand. So please do join us. And if you're watching this later, not live, please do feel free to ask Mary any questions on the comments below, and Mary will follow the post and hopefully get back to you there. So once again, thanks so much for everyone for joining us and listening, and thanks Max for hosting, and well done, Mary. What an incredible job that you have, and really glad that you could share it with us and Strah. All right, everyone, thanks so much. Bye.
Thanks to all of you for the invitation.